We are live now. Fatima, we are live. You can start the session.
Assalamu alaikum all of you. I hope you all will be doing well. So I'm Fatma Fizan hosting this webinar on the behalf of our organizers of today's webinar, which is IEEE Superior University Student Branch. I'm really very thankful to our collaborators, Xperteq, IEEE Independent University Student Branch, IEEE University of South Asia Student Branch, IEEE IUB SB Affinity Group. Thank you so much for joining us in today's webinar. Dear listeners, today we'll be having a great discussion on today's topic, introducing biomedical instrumentation and signal processing lab. BISPL and its research activities, which will be moreover presented by Associate Professor Muhammad Kafiul, who has received BSc in EEE from Islamic University of Technology, Ghazipur, Bangladesh in 2008 and completed his PhD from Department of ECE and US Singapore in the area of neural signal processing back in 2015. Currently, he is serving as an associate professor in the Department of Electrical and Electronic Engineer of Independent University, Bangladesh. His research interests include biomedical instrumentation, signal processing, neural signal processing, brain-computer interface, and many more. So now, handing over this webinar to Mr. Muhammad Kafiul.
Assalamu alaikum. Thank you, Fatima, for the kind introduction. Thank you, IEEE Superior University Student Branch, for this initiative of organizing the webinar, and all the other hosts for supporting this webinar. So I would like to take a moment to present my screen, share my screen. Let me share the slides. I think you can see the slide. Can you confirm if you can see the slide? Because I'm now in full screen and I have
Yes,
the
sir, we
slide in front.
can see your slides.
Oh, okay. So thank you once again for this opportunity to introduce our biomedical instrumentation and signal processing lab at Independent University, Bangladesh. The research project that we have been worked so far, currently working, and some of the achievements. So without further ado, would like to start the presentation. So this is my short biography, which I think Fatima has kindly introduced already. So I'm going to skip that. So we have a very small research group, you know, uh, biomedical instrumentation signal processing at IUB. We started our journey with one single research assistant back into 2016, and then in last year we had almost 6 research assistants working for our lab, and so many students, they have completed their undergraduate senior projects. Some of them are doing their master's thesis as well. And we also have some external collaborator. So this is the Facebook page and the ResearchGate lab. So we found that among all the private universities in Bangladesh, in the area of biomedical engineering, IUB, our current university, it ranks first in Schemago 2022 institutional ranking. And thanks to BISPL, because we are one of the active research group at IUB that are doing research on biomedical engineering. So this is just at a glance the number of research projects that we have got funding for, and some of the students that we have supervised in both undergrad and postgrad level. So let us start talking about what are the basic topics of research of our research group. So as the name suggests that we do research on biomedical instrumentation, biomedical signal processing, all the biomedical and biomedical signal classification as well. So biomedical signal starting from all the non-invasive way that we acquire the biomedical signal from the human body, like EEG, ECG, EMG, EOG, PPG, and so on and so forth. And all the application of these instrumentation to acquire the biomedical signals and then do process the signals and then eventually applying some kind of machine learning or AI based techniques so that we can eventually make some decision in different applications, starting from brain computer interfacing, which is known as BCI, to detect any disease or diagnose any disease, in basic neuroscience research, in human machine interaction, for rehabilitation purpose, for controlling neural prosthetics arm or robotic arms, and some other non-clinical application as well. For example, neuromarketing, biometrics, and entertainment, like controlling smart TV through your brain. So this is the list of the collaborators of our lab. We have a variety of collaborators within IUB, outside IUB in Bangladesh, and also some international collaborators. So we have some of them are very big fish. For example, Professor Syed Sanai from UK. We got our PhD advisor, Dr. Zi Yang from University of Minnesota, and whatnot. I mean, so we are a very humble research group trying to grow, but it's already been like 6 years of our journey. So let us talk about the background on biomedical signals, basically the type of the signal that we work on. So if you take a look at the group name is biomedical instrumentation. So this instrumentation mainly used for acquiring or recording the biomedical signals in a non-invasive way. And then the signal processing and machine learning is applied on the recorded acquired biomedical signals to actually make some decision. For example, to control any computerized device in PCI application, or to understand how the brain works, or for diagnosis or detection of any disease or any particular event that is of interest for different health application or medical application. So the origin of common biological signal, the way that they are named, it is actually the type of the signal, whether the signal can be an electrical signal or mechanical signal, or it can be a mechanical or the sound wave, right? And the organ from which you are acquiring the signal, for example, if it is a heart, then we call it cardio. If it is a brain, then we call it encepha. If it's a muscle, then we call it myo. And then the type of presentation, for example, gram, graphy, or graph or graphy. So graph is obviously the way to present the signal. Gram is actually the picture of the signal or the image of the signal, and graphy is actually the technique by which you acquire the signal. So if we take an example of electrical activity of heart, which is known as ECG, ECG is known for electrocardiography, right? So it is an electrical signal, so electro, and then cardio, and then graphy. So electrocardiography means it is the technique to acquire or record cardiac activity, electrical activity of the of heart, which means the cardiac activity. Okay. So similarly, you can have a signal which is a mechanical type, then accordingly it can be named. For example, if you think about MEG, or magnetic field, magnetic encephalography. So this is a technique to acquire the brain signal through magnetic field. So this is how the name. So in our lab, we mostly focus on EEG, ECG, EMG, and EOG. So these are some of the non-invasive techniques that we can actually design this instrumentation to record this signal by ourselves, and also we can do the processing and classification by ourselves. Moving on, so why it is a different kind of lab to only process biomedical signals? Not only because it has an enormous applications in healthcare and in medicine, but also it is biomedical signals are very different from the traditional signals that we use for communication. For example, the way right now I'm talking, obviously my voice, my video, they are streamed online, right? They are being sent wirelessly. So this communication channel is there. So also there are signal processing, machine learning involved in this kind of the way that we communicate. But if you look at the biomedical signals, one of the most important challenge or difficulties to deal with biomedical signal is that it has a very low signal to noise ratio, or SNR. So this is an example of a graph where showing that x-axis is a frequency and y-axis we have some amplitude, and you can see that noise level, this is the noise level, which is higher than the biomedical signals such as electromyography, EMG, electrocardiography, ECG, or electroencephalography. So that's the point where we have to really be very careful in acquiring biomedical signal and process signal, because our signal of interest is actually merged under the noise. So we have to get rid of this noise, and that is very challenging. And another part is that the biomedical signals are non-stationary in nature, which means that the properties of the signal, they change over time. So for example, the frequency component of the signal, they change over time. And these are also nonlinear, they come from some nonlinear source. So it makes it even further difficult to understand and process this signal or detect some eventful scenario or interesting signal out of this recording. And it has diverse type of noise. And another part is that you cannot use a traditional digital filtering to remove the noise from these biomedical signals, because your noise and your signal, they overlap in both frequency and time domain. So this is another point, that you cannot use simply digital filtering. You have to use some advanced signal processing and machine learning technique to make some sense out of these recorded signals. And if you do not process these biomedical signals well, what happens? You will have false alarm, which means that you will have error in your outcome or in decision making that will mislead the diagnosis, the treatment, or control of any external devices. So as mentioned, the traditional signal processing failed because we have a non-stationary type of signal, and we have noise and signals are overlapping in frequency domain. So let us talk about some of the project that we have been doing, we have done before, or currently we are doing. Okay, so this is the number one project. So this is an example of where we have, basically this is undergoing project. It is not completed yet. Some part of the project has been completed. So this project tells you that you have to have this, skill of instrumentation, of signal processing, of machine learning, and overall integration of the whole devices, all these different subsystems to make a single, single system. So the idea of this project is that if you have people who are diagnosed with quadriplegia or quadriparesis, so these are kind of disease where the person has an kind of paralyzed from neck to toe, all the way from neck to toe, which means it makes them difficult to actually do anything by themselves. So they cannot move by themselves, they cannot eat by themselves, they cannot even touch the smartphone, or cannot use the traditional mouse cursor or mouse. So for this kind of people, for as an assistive device, an assistive system, we plan to develop uh, a, plan to develop a system which can eventually acquire some electrooculography, which means that eyeball movement, the movement of the eye from the subject, because these subjects can eventually move their eyeball, they can blink, okay, but they cannot move their arm. They have lost their motor function totally, okay. So with the help of this eyeball movement, they will be able to control the wheelchair. So that means they can move by themselves, an electrical wheelchair. They can control the mouse cursor, which means that with some limited ability they will be able to use a computer. They can they can control their prosthetic arm. If, let's say that if or an exoskeleton, if their arm is not moving, so they can use that an assistive device to move their arm. Or if they do not have that arm, they can use a prosthetic arm to actually grasp something or place something from one place to another, pick and place. So this is what we have already developed, the EOG recording device, as you can see. So it is an uh, you know example of a biomedical instrumentation to record these signals. So these signals are original signals that are being recorded from one of our volunteers. And we have been able to control individual these things individually, but we have not been able to make a complete system and integrate all of them in a complete package. So this is something that is yet to be done. So this is an example that we have applied some machine learning technique to find out the accuracy, because you have to make the decision which way you should move the electric wheelchair, how you're going to move your mouse cursor, and your prosthetic arm. That means there are some degrees of freedom by which you can actually rotate your arm or move your arm, pick and place some object, something like that. So we have to apply signal processing, you have to apply machine learning to be able to make all this decision. And finally, these decision will be converting to command to control all of these external devices or components. Next project, the application is actually non-clinical application, okay? But the signal that we are recording is actually EEG or electroencephalogram or brain signal. So the idea is that you can, for example, when any ad comes in front of you, for example, you're browsing YouTube, you're browsing Facebook, and then different types of ads, they're coming, sponsored ads in front of you, right? Now, for the ad agencies or for the companies, what kind of, how they will make this ad, whether eventually we are going to like the customer, the target audience, or target clients, whether they will like or dislike these ads. And because based on this like and dislike, they will eventually prompt to buy those products, right? So we can actually understand from the EEG brain signals perspective. So whether you really going to like or dislike your brain signal, I can record your brain signal. I can extract whether you really like or dislike based on by applying some machine learning technique, by extracting some features and applying some classification model, and then I can actually predict your choice. Okay, so this is an example where can be used for neuromarketing, not only neuromarketing but also for any survey purpose, for awareness, creating awareness, and different social awareness creation. This project can be useful. So we have recently got acceptance in an Q1 rank journal, Computational and Intelligent Neuroscience, of this particular project, because we found that this is very important. Here, the application is non-clinical. However, we are still using biomedical signals like EEG. Moving on to the third project. So this is an example when for developing countries like Bangladesh, where not everyone can afford, healthcare service or can afford expensive healthcare services, right? So, this is an example where you can, we have what we have done. We have, you know, made a prototype for a low-cost real-time vein imaging to, draw bloods or for, doing the cannula. So this is a very typical way to do it. But if we can expose, sometimes the veins are not found. Okay, especially for the children, it is very difficult to find the vein when the nurse is trying. So they have to try multiple times to get the prick, and eventually it is very painful for the children. So from that motivation, what we have done, we have simply designed this kind of architecture, and we have placed some led lights so that these veins can be exposed. Okay, and we have used some IR based IR light, infrared light, NIR, most to be precise. We have used some NIR light, near infrared light, to actually expose those veins, so that it is easy to draw the blood or to make any cannula. So this is an example of totally biomedical instrumentation, example of your expertise. We have not used any signal processing here. We have not used any, machine learning here. This is totally, biomedical instrumentation example. And the filter that we have used, they are optical filters. So it is all about some optical filtering and techniques, and make this prototype by ourselves. So this is done by one of the senior project groups. Moving on to the next one. This is an example where we have said that we can use our EMG or electromyogram, which is the recording of your muscle from non-invasively, from the peripheral muscle, from your skin. We can actually record myogram or muscle signals, electrical activity of muscles, and use that to control some prosthetic arm. So this is an example where this prosthetic arm has been 3D printed, designed by one of the senior project students, and they have also, developed the EMG recordings device by themselves, and they have been able to control the prosthetic arm accordingly. And so you can see that they have also used some filtering technique to increase the SNR. So this is an example for the interested students, especially those who would like to do projects on this kind of field, or aspiring young researchers. So there are a lot of things that you can do. So these are the real signal that we have recorded by our designed EMG recording devices. All of these projects that I have shown so far, they have been somehow got accepted in different publication in different venues. Okay. So this is another example, which is a non-clinical example, but we are using biomedical signals. So the title is use of spontaneous eye blinking. So we are talking about eye blinking. So you know that we keep blink. Sometimes we blink intentionally, and most of the time we have to blink unintentionally, right? So we are talking about unintentional or spontaneous eye blinking, and this spontaneous eye blinking can be used for biometric application, for authenticating any human, specific human being, right? So we know that these days, we have RFID, we have biometric authentication, we have fingerprint and all this stuffs, right? But somehow many of these authentications, they can't be made, fake. For example, you can get the fingerprint from your friend and then you can use that fingerprint as if that you are that person, or for example, you can take the RFID card, your ID card from your friend, and then you can use that card. So most of the time, different types of the existing human authentication biometric system, they can be vulnerable, they can be made fake. Okay, they can be exploited. However, I think about spontaneous blinking of yours. So this project actually suggested that we have got accuracy of 98.7%. So we said that we have 46 subjects, each of their eye blinking were recorded by EOG devices, electropholography devices. Okay, and then we found that each of us, or each of them out of 46, they had a different pattern in eye blinking. So the way they blink, they are different. Okay, so their amplitude is different, their timing different, everything. So we have used some machine learning technique, and we found that we can actually identify individual person, all 46 persons, based on their spontaneous blinking of say 5 minutes recording. Now, obviously, this kind of authentication system you cannot use in your university gate pass, right? Because it will take 5 minutes. But think about in sophisticated area like vault, bank vault. We have seen so many movies where the bank vault has been, they have been the robbers. They go to the bank and break the vault and get the money, right? So you need a very sophisticated authentication system in those places. Okay, so here you have to record someone's eye blinking for 5 minutes, and that has to be spontaneous, has to be unintentional, and then only, for example, the lock of your vault can open. Okay, so that's not very easy. That's not very easy to actually copy. Okay, so this is an example where we found that we can do this as well. We can use biomedical signals, but in different non-clinical applications as well. Moving on, the next example. So this is another example of the power of the smartphone that we have been using. Okay, so you know that our smartphone comes with different types of sensors. We have accelerometer, gyroscope, light meter, temperature sensor, you name it. There are a lot of sensors embedded inside your smartphone. Okay, now, but you can see that the elderly people, they have this tendency of getting to they can fall sometimes, and to detect those fall are very important so that you can actually help them when they get fall. Okay, so your smartphone can be used to actually detect this kind of human fall, and eventually the same smartphone can be used to send the warning message to the caregiver. Okay, so this is one of the senior project students that we have done, where we have recorded different fall activities by ourselves, and then we have classified them from fall and non-fall event, and then we got around 92% of accuracy. Again, this is an example where we have not made the acquisition device or recording device by ourselves. What we have done, we just have used the accelerometer of our smartphone to record these event when there is a fall or non-fall, and then we use some classifier in the server to actually classify, to make decision whether there is a fall or non-fall, to detect this kind of fall. Going to the next project. So this is an example where again, the use of a smartphone, you can use your smartphone camera to actually screen basic skin diseases that you have. Okay, so you can use your smartphone to take a picture of your skin. For example, if there is any unusual things on your skin, on your arm, on your leg, on your body, and then you can capture the picture, send wirelessly to a remote server, and then let the server do the classification. Okay, so whether this is this kind of disease or some normal skin. Okay, or whether it is this cancerous skin or not. So you can use this deep learning, machine learning technique to actually classify whether it's normal skin or abnormal skin. Again, the acquisition device is your smartphone. Your camera is your acquisition device sensor. Okay, all we have done is that application of deep learning to identify whether this is a normal skin or abnormal skin. Okay, the lesion of the skin lesion, whether it is normal or abnormal. Another example, again, it's kind of a non-clinical example where you can use this kind of application in smart TV application. Smart TV means that you can actually change the channel by your mind, or the channel can suggest you, the TV will be able to suggest you something that is suitable for you at that moment based on your emotion. Okay, so what we have done, we have been able to use our brain signal, single channel EEG brain signal, to detect our emotion induced by some different types of movie clips that have been shown to the subject. So for example, when there is a horror movie, when there is a relaxing movie, when there is a romantic movie, so these movie clips, the way that our brain signal captured these emotions, okay, by watching these types of movie clip, we can eventually extract brain signal and classify our classifier, we can train our classifier to be able to, detect these emotions by the classifier. Okay, so this is an example where we have got more than 95% on average classification accuracy. This is a very hardcore signal processing technique where we have used some fuzzy logic system for automatic artifact detection and removal from EEG signal. So artifact is known as noise in for the EEG signal. As mentioned, that biomedical signal processing can be very challenging, and especially EEG signal is the most challenging out of all biomedical signals, because EEG signal has the lowest SNR compared to ECG, EMG, or other biomedical signals. So artifact detection removal is one of the open research platform that I have seen this for last 20 years, that artifact detection removal are going on. This kind of research is going on in this field, and yet it is an active and open research platform. Moving on to the next project. Again, this is a typical signal processing and machine learning or deep learning based application where we have used EEG signal classification for application in state SSVP. This is a kind of an BCI, brain computer interface, where we have used convolutional neural network for the classification purpose. So this is too much technical, so I'm not going to go into detail of this one. So what is idea is that in BCI, in brain computer interface, where you can actually control some computerized device through your brain, or it's kind of a mind controlled. Okay, so there are different types of BCI. One is motor imagery, another is SSVP, then we have P300 and so on and so forth. So this is one of the BCI that one of the type of the BCI that is SSVP, which is known as steady state visual evoked potential. So the experiment, what it does, you have the subject is exposed to different types of color images or visual stimulation, where by applying those stimulation or by applying those color stimulation, your brain signal can be changed. Okay, so your brain signal has a different response to different types of images or different types of color. That is known as visual. So your visual evoked potential, that means in the occipital region of your brain, there will be some potential evoked due to the visual stimulation. So that is that kind of BCI. And we have used convolutional neural network, which is very well-known deep learning method to classify any noisy or any big data. This is probably the last one. And this is the most recent one that we have done. So we have been able to screen depression among young adults. So here the young adults refers to age 18 to 25, basically the university students. And we have found that through EEG, by recording EEG or brain signal from the subject, we can actually identify whether the subject has depression or no depression. Okay, so this is a very much recording of the EEG signal, and the EEG signal is validated through basically whether the depression diagnosis is validated through standard questionnaire like a screening tool like PHQ-9, which is a standard screening tool for diagnosis of or screening of depression, which is often used by the psychiatrist. So we have compared these results from EEG recording as well as from the screening tool, and we found that our classifier, like SVM and KNN, they can actually classify with an accuracy of more than 95%. Okay, so this can be in an we're not saying that EEG based depression screening is the alternate method for with the traditional screening, but we are saying that is a complement. You can actually double check whether the traditional system, you can increase the accuracy or the convenience of the traditional system, but if you employ EEG based depression screening in screening of depression or anxiety. Okay, not only that, you can actually improve your accuracy as well, overall accuracy. So that's kind of all that we had in our uh, project that we have been working on. So we have additional projects that due to the time constraint we did not put in the slides. And these are some of the achievements that the publication so far from 2016. So it's kind of a 6.5 years. We have been able to have 47 publication, and these are some of the award that we have received in recognition as a part of this research effort of the small research group BISPL. Uh, so I think that's it. I can stop here, and if there is any question, then we can take.
Thank you, sir, for such a great session. If anyone want to ask any question, you can ask in the chat box. We have a question, sir, from magazine.
Yeah, I think that was more like a comment. Yeah, but we
Yeah,
have a
that was only
question
the
from
comment.
some Tariq.
Uh,
What is the most difficult part of publishing your research? I think what happens that when you work with students, first of all, in our university, we do not have PhD program. We have very limited number of master's program. So most of the work that I have done here is with the undergrad students. Okay. So if you are an undergrad student, you know that, in final year, when you're in the fourth year of your undergrad engineering undergrad, then you have only 1 year to do your final year project. And then once you graduate, you basically disappear from the university, right? If there is no research assistant position or something like that. So we have very limited fund. uh, uh, so what the most difficult part of publishing the research is that student finish their project, but then they graduate, and obviously I cannot uh, get hold on to them. So go out, they graduate, and they get job, or they go for higher studies, and we basically lost the connection between the students and myself and our group. So I have to basically, sometimes I have to write the paper by myself based on the student research. Okay, so that is when you have a very stable research group, where there are PhD students, postdocs, research engineer, research assistant, research associates, very good team. Then you can actually employ others to, or you can divide the workload among others. But when you do not have that much funding, so that means you cannot, uh, you know, uh, recruit research assistant or paid. So if it is a volunteer, then voluntary work, then it will not go further, right? Not much work can be done with a voluntary association. But anyway, I think still that what we have achieved with the help of the undergrad students, it is something significant as far as the location or the resources is concerned. With the limited resources, limited funding, I think that is something uh, not bad. I hope that answered the question.
Is there anyone else who want to ask the question? You can ask the question. We have another question from Amir Hamza. Uh,
Basically, if you are an IEEE, member, student member, then you can apply for some fund from IEEE. Okay, in your region, in your chapter, in your section, there are some very limited amount of fund available for IEEE. Those students are IEEE members to trying to do research. But eventually, what happens? Your professor, who should be the PI, principal investigator of any research project, they are supposed to apply for grants because as a student, you cannot get fund by yourself directly. Okay, because you cannot manage a research project by yourself. If I mean, given that if you are a student, uh, because you have to have a PI, principal investigator, to apply for a grant, apply for a research project. Okay, and that can either come from the university or from other funding agency, or some can also apply for international funding. But given that you are a professor yourself or you are a PI, you can be a PI. If you are a student, then you have to ask the professor to manage a fund for you, or you can directly apply to IEEE. There are some funding for example, if you want to do some humanitarian projects, there are some small amount of fund available in your section or in your region. For example, we are in region 10, right? Asia Pacific region. So in region 10, there are some specific funds. Call for funds can available in your IEEE website as well. You can explore those opportunities. Yeah, I mean, for high impact journals, obviously, you need a very good student or assistant who can write, who have very good scientific writing ability. Otherwise, it's difficult. So most of the publications that I have shown here, with undergrad projects, I have been able to publish in conferences. But the journal paper, that publication that I had, it was actually collaboration between our university and some other universities with some other professors, even from international. How can we proceed the publication within biosignals and authenticate the results we achieved? I'm not sure what exactly is the question here, but I believe that for the students, with if you have limited resources and limited funding, so what you can do actually, you can first of all, if you have a systematic way of research funding or research project, then through that project you can get an approval from your university, so that you can work with subjects like human subjects. For example, all the project that I have shown here, I had to get the approval from my university, which is known as IRB, Institutional Review Board, similar kind of authority. That authority will authorize your research project, that yes, you can actually do these projects, or you can record the biosignals from human subjects, for example, with your friends as a students, or the faculty members in your university, or whatever, whoever the people or human subjects that you have to get approval. Okay, and then once you have these biosignals recorded, let's say that you are applying some signal processing or machine learning technique to make some decision or make some outcome out of these biosignals. So then you have to compare your results with already existing literature, right? To prove that, let's say that you have uh, proposed a new algorithm in signal processing or new machine learning or deep learning model, or you have said that you have proposed a new system to acquire these signals with high quality. So either way, whatever you are going to claim, you have to prove that, you have to validate those against some standard technique or against the existing works in literature. Okay, so let's say that you have designed an EEG recording device by yourself, so you have to validate that with already approved EEG recording device available, commercially available. So this is how you can validate your results with an existing tool, already approved existing tool, or you can compare your results with the results of others who have already published in the literature in in peer-reviewed journals or conferences. This is how you can actually publish. Yes, you can get the slides. I've already shared with the host. I hope that they can share with them, with all of you.
If anyone else has any question, you can ask.
Yeah, I hope that it is helpful. And especially my webinar, this talk was designed for the students so that they can know what is the world of biomedical instrumentation, signal processing, and where they can actually contribute. So you can see that all of these research, it required the basics of EEE and CSE. So if you are from EEE, CSE background, then you can actually work on these areas. Future plan is, I mean, I need more funding so that I have a stable research group, and then only good quality research can possible. I need funding. So I'm applying for different, I'm trying to apply for different opportunity to get fund. Thank you.
If anyone have any question, you can ask. Otherwise, we can end the session by thanking our host.
So one last thing that, uh, I think what I have presented here with very limited number of resources and very limited number of opportunities to research compared to in developed work, uh, I have completed my PhD from NUS, National University of Singapore. So I have seen already that what is the typical traditional research trend in those developed countries. Compared to that, we have almost nothing. But still, I think there are different ways to learn. I mean, I'm not saying that I'm doing very high end research, high quality research, but what I'm trying with the students is actually to motivate them. uh, Actually, it's a learning phase for the students as well, especially those in the undergrad level. uh, because original research, you cannot do very good research without PhD program if you do not have a PhD students. Okay, so but with the limited resource and funding, it is actually possible if you want. If you want, then so many things can possible. And obviously, you have to be very open-minded to make some collaboration. All of some of the good quality journal that we have published, it was not possible without collaboration. So collaboration is very important. And those who are in IEEE, I'll tell you that, as an IEEE member, you can have an international mentor as well. Okay. For example, an IEEE fellow can be your mentor through IEEE Collaboratech platform. So you have to explore these opportunities given by IEEE. You can have a multinational or international collaboration as well, being a student. But you have to explore. You have to be brave enough to, for example, contact a professor, an international professor, or to get some help. Okay, for a particular project, you have to be brave enough to get this network. So networking, communication skill, some of the and team bonding. So this is something that very important for research these days. Nobody does research isolately in a single room, or in a lab far away from the city. It's not possible. So I think that the students who are listening to me, my sincere advice to them is that get to know each other, make some connections with the similar type of research interest, and then take help from others.
Thank you, sir, for giving your time to us. We are really happy to have you with us. You give us a lot of information which we were expecting on this. Thanks a lot, sir. Once again, you gave a brilliant session today. Just amazing that is.
Thank you, Abdullah Marov. Thank you, Fatima, all the hosts and all the collaborators for organizing this webinar and all the audience for your presence. Thank you so much.
Thank you,
Thank you
sir.
for this
Thanks a lot,
opportunity.
sir.